Good afternoon, everybody. I would like to thank Paul for this invitation because it's a great opportunity to present our work using immune cell methylomes to determine blood cell subtype heterogeneity by a deconvolution approach. This, okay. Why to do a focus on animal health today? It's because the livestock is one of the pillars of the global food system, and now, however, it's submitted to global environmental changes, such as heat stress, with modification of water and nutrition resources, and with increase of exposure to new pathogens. And in this context, the health of farm animals is very important to maintain the welfare and metabolic homeostasis, their efficiency and their fertility, and to have a good production. The animal health is dependent of immune capacities, and the immune capacity is defined by the number and the activity of hematopoietic cells. Hematopoietic cells are classified in several subpopulations, originate from a long and complex process of differentiation, and this process takes place during the fetal life, display a maturation period just after the birth, and produce differentiated cells throughout the life of the animal. And this process needs interaction between transcriptional factor activities and epigenetic marks with a very spatiotemporal specificity. Then each cell type is characterized by a specific gene expression pattern supported by a specific epigenetic state as well as a DNA methylation profile. And the evaluation and follow-up of the immune capacities of animals need tools, the most informative, the lesser invasive, and easier to routinely use. Currently, the blood formula was performed to quantify the three major cell subtypes, lymphocyte, neutrophil, and monocyte. But this assay needs fresh blood sample freshly processed with transport of sample and access to calibrate automated analyzer. And we propose, in fact, a new tools to use biobanked blood samples from experimental and commercial farm assays, for example, because these samples are rich and available sources of genomic DNA with a good integrity. And we propose to perform the pangenomic DNA methylation profile to explore the cell subtype proportion by deconvolution approach with references. Jessica have introduced this method this morning. It's a very clear introduction. Sorry, je me suis trompée. And to perform the deconvolution approach with references, we have three steps. The first is the identification of immune cell subtype methylomes by reduced representation bisulfite sequencing. The second step is identification of more discriminant position by multivariate analysis. And after, and finally, to calculate the relative leukocyte proportion using EpiDish algorithm. This algorithm was developed from a human study. We use a very particular experimental design because we limited the genetic variation because we use clone animals on all these animals have exactly the same genotype, and we added some cow with variable genotypes. We limit also the physiological stage effect because all animals were submitted to hormonal synchronization, and the sampling was performed at day 15 of the cycle, and we limit the environment effect because whole animals were in the same breeding. From the blood sample, we perform a purification of PBMC, and after purification of the 3 subpopulation of leukocytes, monocytes, lymphocyte T, CD4, and neutrophil. Using RRBS, we identified more than 1.3 million of CPG with a pertinent genomic distribution. And using the methylation information of this CPG, we perform PCR. And as you can see in this slide, the PCR clearly separates the 3 different subpopulations, and with a clear separation between the 2 lineage, lymphoid and myeloid lineage. After that, it's possible to perform some differential analysis by using MethylKit software, and we use the option to eliminate all positions with a methylation differential of less than 25%. And by this way, 6,300 differentially methylated cytosine were identified and used to conduct PCR or PLSDA selection to identify the more discriminant position. With this, it's possible to identify 146 discriminant position, and using the methylation information of this very small subset of discriminant position is possible to have a clear separation between the 3 subpopulation. With and the more important is that the 2 first axes of PCR explain more than 96% of variability between the samples. The heat map display this clear separation between the 3 cell subpopulation, and display also a very specific methylation profile for each cell subtype. This discriminant position have not non-random chromosomic distribution. For example, there were absent of the chromosome 27 or chromosome 28, and we have one bigger cluster in chromosome 19. And there have also non-random genomic distribution, and the most interestingly is 83% of them target genes, and we target more precisely 81 unique genes. And by enrichment analysis, it's possible the enrichment analysis revealed, in fact, 3 common functions and pathway related to regulation of transcription, the interaction with RNA polymerase II, and with factor with DNA binding. And the second function is related to component of cytosol, cytoskeleton, and cytoplasm. It's a very intracellular components. And the last is in relation with the signaling pathway. such as protein phosphorylation and protein kinase activity. We look at the stability of these methylation profiles of the whole discriminant position using new purified monocytes. We added 23 new monocytes from other animals or from other physiological stage. This is from lactation stage. And you can see here all monocytes clustered together, and we observe a low variability of methylation profile between this cell type. We look also the methylation profile in heterogeneous cell population. For that, we prepare Hélène PBMC preparation, and we use also 21 blood DNA samples. And the heat map display the conserved clustering with here the neutrophile and here all the monocytes, and all the PBMC are in orange, in orange in this part, and the PBMC are closely clustered with lymphocyte T CD4, are coding to expected high proportion of lymphocyte T CD4 in the PBMC fraction. And all the blood sample are here and cluster together and display a very specific methylation profile according to mixed cell subpopulation. Concerning the deconvolution approach, we calculate the relative leukocyte proportion using Epidish algorithm, and using the methylation data from the whole PBMC libraries, we predict the predicted leukocyte proportion are close to expected proportion in PBMC fraction, with a high level of lymphocytes and a very low level of neutrophil, because the neurophiles are eliminated by negative selection during the centrifugation on fecal gradient. And using methylation data from blood libraries, we can compare the leukocyte proportion determined by the conventional blood formulation in this, and the relative predicted proportion in this axis. And as you can see, the Pearce correlation is very good. And we have a good prediction with a low monocyte level and a high level of granulocyte and lymphocyte. But at the cell subtype level, for us, the correlation is not very nice, because for all these three different populations, the Pearce correlation is decreased. In conclusion, we have specific methylome for 3 immunocell subtypes. We have identified a small subset of 146 discriminant positions, and the deconvolution approach using references allows a good determination of relative proportion of the 3 subpopulations as observed in PBMC. But the prediction lacks precision at the cell subtype level as observed in blood sample. And to improve the deconvolution approach, we propose first to increase the number of cell-specific positions to have a better powers, and increase also the animal number and the genetic variability of the next experimental design. Thank you for your attention.
Really nice presentation, and probably a solution to some problems that we are facing. So I wanted to find out if you think this, your findings can be extended to other cell types, like not from the blood.
I don't understand. So,
I'm asking.
I think this is just because I can.
Okay, I'm asking if you think your findings can be extended to other cell types.
Yes.
Not cells from the blood, but like mammary gland tissue or milk somatic cells.
Alors, first is to extend to lymphocyte B and lymphocyte T, CD8 and NK cells, as Jessica looked at this morning. And I think it's also important to have some methylomes from activated immune cells with specific inflammation or infection. And it's a first in blood.
Okay. Thank you.
This is probably a crazy idea, but would it be possible to build a chip that was capable of measuring RNA or cDNA as well as DNA methylation changes? You know, there's probably going to be a limit, I would think, to the ability of the epigenome alone to distinguish cell types. But if, like I say, this is probably crazy, but if in the same sample you had information about RNA, would that increase the accuracy of your ability to do deconvolution? Like I say, it's probably a crazy idea.
I think it's yeah, yes, sorry, it's quite interesting to have the two approach in using RNA and using DNA and with the convolution approach, and it's possible to have a good interaction between methylation and methylation variation and gene expression variation. But in this, for us, one the more interesting is the application, because we can use it with biobank and blood samples, and it's more difficult to perform RNA approach with this type of blood samples.